You can really split gene transfer into 3 steps. So step 1 is that you get the DNA, one plasmid from your vector, right? So that would be, for example, a virus or something. And you also have from a human whatever segment of DNA you want. So when you have that, what you do is you take a restriction enzyme. I pick one which is ECO-R1, which is ECO because it's found in E. coli, and R1 because it's an R strain, it's the first one they found. And you just take it and you cut the DNA there. So I think ECO-R1 is this sequence, which coincidentally is palindromic, which is because this enzyme is a dimer, so that's why it reads both ways. So anyway, basically it's going to cut, I think, right? If I made a mistake. So really, it just cuts at that site, and you have the 2 sticky ends, right? And then, as you learned before, ligase is the enzyme that comes and will connect together. Although I guess technically the step that comes first is that you put PCR, so you would have a whole bunch. That's what. Oops. Wow. I forgot I did that. Okay, no, but basically, sorry. Point me that the PCR should happen first, and then you use ligase to connect all together. So that's an inaccurate order of things. But after you do that, if it's prokaryotic, if it's prokaryotic, you have something called transformation. So it's going to basically take the plasmid into it. And if it's a normal, like some people use what the example is a sheep. They have the goat. They produce silk, so that would be a different process. But basically, you get protein out of it, or you want to collect whatever products. And I guess since I talked about epigenetics before, I think there's like a just quick tie that's kind of interesting instead. So ECO-R1, basically the reason why it's found in bacteria, like the reason why bacteria develops this gene or whatever, is because viruses attack bacteria and they insert their DNA or their genome into those bacteria. So in order to prevent it, what they did is that they have something like ECO-R1, which is going to cut out DNA out of it because it recognizes a certain sequence, and this sequence occurs about every 1 in 4,000 bases. So it does this to protect itself, and then the mechanism is that you have ECO-R1, which will cut DNA, but then in the original thing, it also has another enzyme called methylase, I think, I'm pretty sure methylase. And methylase just constantly, whenever it sees that same site, the same GAA-TTC, it's going to lay down a methyl group, and that will prevent the ECO-R1 from cutting at that site. So basically, it has 2 mechanisms in competition, in which one is methylating its entire genome, and the other one is cutting whenever there's no methylation. So that's how they can make sure they only cut out foreign DNA. And then the fun thing, though, is that like if by any chance, because this is kind of like a race between these 2 enzymes, if on every once in a while the ECO-R1 will, the methylase will get to the viral DNA before ECO-R1 does, and therefore it's going to methylate it before it has a chance to cut out and recognize that it's epigenetic resistance. So it's not actually genetic, so it doesn't have to actually learn how to combat antibiotic or something. It's just epigenetic. So once it leaves the organism, yay!